talking to? Just here's fine. Can everybody hear me?
Yep.
Okay, good. All right, thanks. Thank you very much, Tom. It's a pleasure to be in this very interesting meeting. I haven't attended one like this before, and I certainly have learnt a lot. However, permit me to share some of my own feelings about what the genesis of colorectal cancer is and what we can do about it. I'm a practicing gastroenterologist. I'm not an epidemiologist, but I'm honored to be in the session because I have a lot of respect for them. It's been noted for a number of years in epidemiological studies looking at the rate of colon cancer in different countries around the world and by how much it varies, even up to about 50-fold. What's been interesting then is that people have coupled this with the dietary inquiry in those different countries and tried to tease out certain characteristics of the diet that might be associated with high or low risk. And now there's substantial, it's even considered convincing evidence by the world authorities that red meat, and particularly processed meat, are associated with increased risk, whereas fiber is associated with a decreased risk. We've already heard that there are many other factors also associated with cancer risk, but these are the predominant things. And the great thing about it is that we can do something about these. It was calculated by Doll and Pito a number of years in Oxford that over 90% of all GI cancers were due to differences in diet. Now that's obviously a speculation, but it actually is based on pretty extraordinary evidence. And the question of meat has been amplified again by the World Health Organization just recently, warning people that meat is a primary carcinogen. So let's start off with fiber. How might it actually prevent colon cancer? Fiber is basically a complex carbohydrate in the diet derived from plant sources that escapes small bowel digestion and thus reaches the colon. And there it provides food for the microbiome. It was first attributed to the low risk of colon cancer in Africans by Burkitt over 50 years ago. He basically went around with a pen and pencil and noted what people ate and what their disease patterns were, and he linked the two together, and it's basically a classic study. More recently, the EPIC study in Europe has estimated that there's a 40% risk reduction in populations with low dietary fiber that double their fiber intake. So it appears that there is some way of actually changing risk by dietary intervention. Red meat is the big culprit that people are worried about at the moment, and it's quite unique. Apart from being absolutely delicious, it increases luminal carcinogens through its heme content and the microbial metabolism of protein residues to things such as nitrosamines, aromatic hydrocarbons, and phenols. And particularly if you use barbecuing, then you generate a compound called heterocyclic amines, which is very carcinogenic. And again, in an epidemiological study, there was a 35% increase in CRC risk when more than 160 grams per day of red and processed meat intake was compared to less than 20 grams per day. In addition, the increase in CRC risk associated with high red meat and processed meat was higher in the group with low fiber intake than in the group with high fiber intake. So now we're beginning to see that other dietary components can modify the effects of others. And this is important to know because we don't just eat meat, we eat a balanced diet. In fact, we are omnivores, and we therefore should be able to tolerate a balanced diet. And basic studies have shown that the proliferative effects of heme on rat colonic mucosa can be prevented if spinach is taken at the same time. And in humans, resistant starch reduces the proliferative and oncogenic effects of a high red meat diet, shown beautifully by a study of Humphreys in Australia recently, where they fed 300 grams of meat a day for 4 weeks with or without resistant starch, and they showed that the oncogenic effects of the high meat diet were actually prevented by the resistant starch. Fat is another culprit, and it's interesting because it's more of an indirect mechanism. We've already heard that there's an association between obesity and colon cancer through its inflammatory effects in distal parts of the body. But it has a direct effect in the colon, mainly due to its effect on hepatic synthesis of bile acids. And the more fat you eat, the higher quantities go into the gut, and a proportion escape reabsorption by the small intestine and go into the colon, where they're metabolized by specific microbes to produce secondary bile acids, which in experimental situations are highly carcinogenic. More recent study has linked taurine bile acids, which is basically taurine is a sulfur-containing amino acid, but it's conjugated within bile acids. And if in fact you can increase the amount of taurine bile acids going into the colon by feeding saturated fat, you produce a blossom of a specific microbe, Bilophila wadsworthia, which produces hydrogen sulfide, which is strongly genotoxic. And then furthermore, saturated fat has inflammatory actions mediated through prostanoid metabolism. So there's always the possibility of some dietary fat escaping digestion and entering the colon. So I'm flipping backwards and forwards. Fruit and vegetables are beneficial. Basically, they suppress colon cancer due to their antioxidant content and anti-neoplastic effects due to their contents of vitamins C, folate, for instance, micronutrients, selenium, calcium, and bioactive phytochemical compounds such as phenolics, generally categorized as phenolic acids, flavonoids, stilbenes, coumarins, and tannins. And what's very interesting about phytochemicals is, again, in order for them to become active, you require the microbiota to convert them or to break down cell walls and release them at a local level in the colon, where it can become biologically active and produce these suppressive effects. So there's always been a lot of argument, well, you know, is colon cancer genetic or is it environmental? And this is probably the best and strongest study, epidemiological study, that shows that it is highly environmental. And it's Limeshan's study of Japanese migrants to Hawaii at the turn of the last century, where at that time the risk of colon cancer in Japan was extremely low on the traditional Japanese diet. It's changed dramatically now. But within one generation, let's see if I can get this marker going down here. No, it doesn't work.
Working.
Is it working? Oh, there it is there. Yeah, so this is their low rate when they're in Japan. And then after one generation, it's right up here, which is a level actually slightly higher than native Hawaiians. And basically it's associated with westernization of their diet. The same pattern occurred with breast cancer, but it was delayed to the second generation, and there it's thought that the change was more related to epigenetic than specific dietary changes. So from this we know that colon cancer is a westernized disease. That picture that I showed you at the beginning shows that all of the high incidence areas are in people consuming a westernized diet. So what do we know about westernization? It's been associated with dramatic increases in lifespan from less than 25 years before the Industrial Revolution to over 80 today. So everybody will say, well, you know, westernization is fantastic. The problem is that those people getting through now are now getting other forms of disease which are impairing quality of life. This change is too rapid to be attributed to genetic evolution that takes millions of years, but it could be to the remarkability of humans to adapt to their environment and the ability to avoid life-threatening events such as perinatal complications, acute infections, trauma, and war. And it's now been considered that Westernized diseases, which include colon cancer, present the most serious threat to public health in the USA today. So we've got to do something about it. Now, what do we know about fruit? From what we know about the process of evolution, the dietary needs of every organism are genetically determined. And some have argued that our health would be better maintained by the diet that first established Homo sapiens in the Paleolithic era in Africa, a period that lasted from 2.5 million years ago to 11,000 years ago. Furthermore, coprolite analysis of fecal residues in cave dwellers in southwestern United States have shown evidence of high grain consumption. And a very fascinating study that I saw on, again, dental remains of people going even back before this bacillus and before Homo sapiens, Homo habilis, and looking at dental isotope contents and showed that evidence that our predecessors were actually grazers and ate very little meat at all. So what's wrong with the Westernized diet? It's grilled, it's fried, it has a lot of fat, it has a lot of simple carbohydrate. We usually wash it down with a glass of beer. It's full of preservatives. I come from Pittsburgh, and that's where Heinz comes from. Where are the fruit and veggies, and where's the fiber? And, the problem is that this is delicious. I mean, and you can feed this to anybody around the world, and they would love it. But, that's food technology, and we have to try and compete against that, which is difficult. So the hypothesis that we've built is that it's not the diet, but it's the microbiome metabolome that is responsible for colon cancer. So just to take you through, I know this is a very mixed audience, and give you some sort of idea of what I'm trying to talk about. This is a simplified version of the adult or even childhood GI tract. At the top here, we have the small intestine, and then at the bottom, the bowl area down there is the large intestine. And we eat food, it's digested. Most of it, in fact, is absorbed as amino acids, glucose, fatty acids, vitamins, which are essential for body health. However, a certain proportion of carbohydrates, particularly if they're complex carbohydrates, and proteins, form residues and go through to the colon, where they're basically fermented by the microbiota. And if you eat a high carbohydrate diet or high fiber containing diet, it's mainly saccharolytic fermentation, and it produces a number of things called short chain fatty acids. And not by chance, butyrate, one of those short chain fatty acids, is the primary energy source for the lining of the bowel. It is the thing that maintains the health of the colon. So you can immediately see there's a symbiotic relation or mutualistic adaptation that's gone on. If, however, and also if you eat a lot of fiber, it generates vitamins such as folate, biotin, and then there's polyphenols that I talked about before, which are released at a local level, and it maintains mucosal health. If in fact you have a high protein diet devoid in fiber, such as meat or fat, then you get generation of toxic chemicals such as hydrogen sulfide, bile acids, and ammonia, which are inflammatory, and we heard from the previous speaker that increases your risk of colon cancer. That's very simplistic. It's actually far more complex than that. So I thought you'd all like to drop off to sleep now before I go into this metabolic pathway. But this is what really happens inside your body after you have a meal or whilst you're living, basically. And it's an integration of the enzymes of bacterial groups identified by microarray analysis, host metabolic enzymes, and fecal and urine metabolites by NMR. And hidden right in the middle here, this thing goes here, is the Tcl Krebs cycle. So Krebs cycle we thought was metabolism. It isn't. It's a minor part of, the starry universe. And I just give you this to show where we're heading. I'm not going to go into much more detail about it at the moment, other to say that, we're entering a very interesting field. However, we have identified some good and bad microbial products, which I've listed here and which I'll talk about as we go through. And really, what determines who gets colon cancer and who doesn't is whether there's a balance between the good effects and the bad effects. So underlying all of this is your genetic susceptibility and, who gets a polyp and who doesn't. The process can be, in fact, arrested if, in fact, you change your diet. This is saccharolytic fermentation for those of you who like chemistry. I'm not going to go into the remarkable effects of butyrate, but butyrate is one of those short-chain fatty acids that I talked about. And it's not only essential for nutrition of the colonic cells, but it also has a multitude of other factors which control the turnover and the anti-neoplastic effects of your diet. So it has apotopic effects through a whole lot of mediated reactions, which I'm not going into now. It reduces inflammation. It has metabolic effects. And a new thing that's becoming more and more identified now is that it actually prevents the action of carcinogens such as secondary bile acids or tobacco or alcohol, and so therefore can prevent the effects of meat on colon cancer risk. Fruit and vegetables, interestingly, have very similar effects to butyrate and short-chain fatty acids, and they have the same anti-inflammatory and anti-carcinogen effect at a mucosal level in the colon, so they have to be released locally. And some studies we did early on, we actually measured colonic content, so we evacuated the colons, and this was a study in Africans and African Americans, and we found that basically here we've got the dietary intakes in yellow and the colonic evacuates in blue, and you can see the blue quantities are far higher than what you're eating in the diet, indicating that the microbes are synthesizing these vitamins here, which are all essential for DNA repair and prevention of neoplastic change. So now I enter the meat of my study, which is basically looking at Africans and African Americans. So I had the privilege of working as a gastroenterologist in rural Africa, and we did colonoscopy for people bleeding and what have you, and it was usually associated with infections and things. We never ever saw polyps, nor did we see diverticulum, as I mentioned earlier. And then I came back to the States, did colonoscopies in African Americans, and 50% of them you could find a polyp. So why is this? What's the cause for it? So we got little grants, first of all, and then it progressed to bigger grants to actually compare the dietary differences that I've talked about before. So we looked at small groups because we did intensive studies. We did high risk, 50 to 65-year-olds. We looked at diet, colonoscopy findings, fecal and colonic microbiota, fecal and colonic metabolites, and most importantly, we did mucosal biopsies to look at inflammation and proliferation biomarkers of cancer risk. And it's been shown that, obviously we're doing short-term studies, you can't wait for cancer to appear or not appear. I'll be dead in my grave by that happened. And so you do the next best thing, which is to look at changes in the mucosa which predict the transformation of normal mucosa to neoplasia. And so we did rural studies in Africa, first of all, looking at their diets, doing dietary inquiries, and basically, you don't have to be a nutrition scientist to see that the diets were completely different, how they were prepared and how they were eaten. And here, this is mainly the staple diet, which is maize meal, cornmeal, which is stirred up into a porridge and eaten commonly throughout the day. No frying involved here at all. And here's our delicious American diet here. And basically, if you look at the dietary analysis, Africans ate a third amount of meat and fat, and 3 times as much fiber, which was tying into the idea of what might protect and what might exacerbate colon cancer. And the colonoscopic findings were amazing because, as I said before, polyps were found commonly in African Americans, 9 of 20, and diverticular in 14 out of 20. African colons were characterized by a lymphocytic colitis in 7 out of 20, and parasites. No polyps, no diverticular. And this was an unexpected finding. The gastroenterologists in the audience here will see this, and there actually were areas that looked like aphthous ulcers, and I thought, gosh, we're finding Crohn's disease, which is supposed to be extremely rare. And when we took biopsies, even the normal mucosa in between these areas showed intense lymphocytic infiltration, as shown here. It's not actually projecting very well here. And the occasional schistosoma and granuloma. And occasionally we would come across segments of tapeworm as well. So, we expect that this was, or suspect this was all due to chronic parasite infections. And the reason that it doesn't progress to cancer like the other situations is because we found very high rates of butyrogenesis. And this puts it graphically here. This is Caucasian American, African American, and an African. And I don't know how well it shows over there, but in the African there's 1 or 2 proliferative cells in the crypt, whereas in Caucasian Americans and African Americans there was staining basically throughout the crypt. And when you show it in, it's even more dramatic in bar form. So this is the joys of doing GI research, is that, we didn't only collect colonic sample or fecal samples, but we evacuated. And when you do a colonoscopy, people throw that away. We collected it assiduously and shake it up and take samples and therefore get an idea of what's happening in the total colon rather than just in a fecal sample. And when we looked at the microbiome makeup between the two groups, it's completely different, as again you can see without being a microbiologist. So this was 12 individuals, 1, 2, 3, 4, 5, 6, 7, 8, 12 African Americans, and then 12 native Africans here. And you can see that the African Americans are dominated by Bacteroides, whereas Africans are dominated by Prevotella species. And this is at a genus level. Containing large numbers, Dialister, Oscillospora, Succinovibria, and Zelenovibacter, which are all associated with breakdown of starch and butyrogenesis. And when we looked at target analysis, we found indeed that Africans had higher levels of saccharolytic fermentation with high rates of butyrogenesis, whereas African Americans, on the other hand, high levels of secondary bile acids and bile acid synthesizing bacteria, so reciprocal changes. So how can we be assured that these differences in colon cancer was not due to other environmental factors such as food storage, preservation, sanitation, housing, electricity, public, because they live in completely different situations. And so to get a better angle on this, what we did is that we designed a unique study where we switched diets. Sorry about that. And what we did is, first of all, we studied 20 Africans in their home environment. We visited them at home, measured what they ate, and did all of our testing, as I've described already. We did the same for African Americans. And then we admitted them to an not hospital, in institution, to actually cook the meals and give them the meals and make sure everything that we gave them was consumed. Because the problem with dietary studies generally is that you say, oh, go home and eat a high fiber diet. They'll do it for a few days and then forget about it. And so this is proof of concept. We really want to see whether or not changing the diet can change the microbes, the metabolites, and the mucosa. And so we put African Americans in the CTRC. That was easy but expensive. And in Africa, we found a disused lodge, and we took that over for 2 weeks and cooked them all their meals and measured what was eaten. And the results were absolutely spectacular, and they were recently published in Nature Communications. And the essence of the dietary change was basically native Africans were given a westernized type diet, so they were given a high-fat, low-fiber diet, whereas African Americans were given the African type diet, which was low-fat and high-fiber. So we're going to focus on those two things. And this shows here that our primary endpoint is looking at proliferation biomarkers of cancer risk. And when Americans were given the African diet, you can see the proliferative cells decreased dramatically. And when Africans were given the delicious westernized American diet, which is hamburgers and all those lovely things, proliferation, suddenly began to appear within 2 weeks. And it's shown dramatically here in bar form. We also looked at inflammation as measured by intraepithelial lymphocytes and by macrophages, and showed again the reciprocal changes, suppression in African Americans and increases in native Africans. We looked at our targeted analysis for butyrate production, and indeed in African Americans we were able to induce butyrogenesis, whereas the reverse occurred in Africans given the westernized diet. Bile acids followed the same pattern. So in a baseline, African American bile acids, secondary bile acids were very high. They were dramatically suppressed after intervention, and in Africans they were increased in the westernized diet. And impressively, I said before that we collected the total colonic contents and measured what was happening there, not just in fecal samples. Africanization of the diet increased the quantities of butyrate and total colonic evacuates 2.5 times, whereas westernized reduced them by half. And on the other hand, Africanization reduced colonic secondary bile acids by 70%, and westernization increased them by a massive 400%. So these aren't just little changes, they are massive changes, and all induced by a change in diet in 2 weeks. So there's an enormous amount of information in this, because we looked at the whole plethora of different microbes that are contained within fecal samples. But we picked out some just to illustrate to you at the moment. We've talked about butyrate producers. If you produce a lot of fermentation, you produce hydrogen, and you have to get rid of it. And indeed, we showed that, in fact, if you increased hydrogen, the hydrogenotrophic organisms mopped it up and increased. And there's a lot of interest in biophilia at the moment, following the Dakota's study. And we showed that, indeed, in African Americans, when they were given the African diet, there was a suppression of biophilia, specific bacteria associated with inflammation. Whereas Fusobacterium has now been associated with colonic tumors, and there's a microclimate now which has been shown in solid tumors throughout the body, which contains its own microbiome, and Fusobacterium is one of those organisms. And indeed, it was increased in Africans given the westernized diet. So just another bit of systems biology that we're going into at the moment. Rather than go through each individual microbial network, this gives you an overview of what we're doing. So this is basically here complex interactions between different microbes doing the same thing, either increasing together or decreasing together. And this is African Americans on their basic diet, westernized diet. We give them an African diet, and look at all these extra pathways that open up here. And all of these extra pathways are associated with butyrogenesis. And exact reciprocal thing happens in native Africans. At baseline, you can see there's a wide spectrum of metabolic activity. You give them a westernized diet, and the whole thing's suppressed. And this shows it basically through the NMR spectra, and this is Africans at the bottom here, and just look at it here. This is a bit difficult to follow. This is home environment and then dietary intervention, and you can see that there's a lot of metabolic activity in Africans on their home diet. Put them on a Westernized diet and it basically all disappears apart from choline, which is induced and is associated with atherogenesis, but that's another story. And the exact reciprocal phenotype was shown in African Americans. This going back to my nice interaction study. Again, I'm not going to expect you to go and follow this, but look at the two panels at the bottom here. These were, the lines here show significant changes in Africans after Westernized diet and African Americans after Africanized diet. And you could basically see one flips one way and one flips the other way. So we've got a highly resilient dynamic system, which in fact can be modified by dietary change. Just to end, the studies we're trying to move on to now is we're really concerned about the extreme risk of colon cancer in Alaskan Native people, over 100 per 100,000. Somebody earlier said that Australia had the higher rate. Actually, you need to amend your comments on that. And you know, we all thought they had a great diet because they eat all the fish oils and so on, but unfortunately the fish oils have got a lot of carcinogens associated with smoke products as well, high meat carcinogens as well, but most importantly, they're low in fiber. They eat hardly any fiber at all. And this shows that, you know, this is about my only slide which mentions problems of youth, and it's for this reason that screening is indicated over the age of 40 in Alaskan Native people, because as you can see right down here, you know, there's significant increases in the risk compared to US whites, and it goes right throughout the whole of lifespan. And we did a pilot study looking at fecal bile acids because we're interested, as we mentioned before, in short-chain fatty acids, and indeed demonstrated that native Alaskans had the highest rates of bile acids of any of the groups that we've looked at, including African Americans. And when we looked at short-chain fatty acids, again, they were dramatically lower in Alaskan natives than the other two groups, particularly native Africans, indicating that there probably is a fiber deficiency. And so the idea is that basically we depend on a balanced diet and they're missing a whole chunk of it. If you can restore this by restoring high fiber foods, you should in fact be able to reverse the whole situation. So, Mr. Chairman, I'd summarize by saying the microbiome behaves as a community wherein intermicrobial interaction strives to produce a metabolic phenotype that supports colonic health and function. It has a genetically determined need for food residues derived from a healthy, balanced diet. The provision of an imbalanced diet leads to disturbance of structure and function with the unopposed production of metabolites that can induce inflammation and proliferation, which increase the rate of neoplastic change. And we would conclude that from epidemiological studies, we know that Westernization of the diet leads to an increase in colon cancer within one generation. But our results show that a change of diet composition produces immediate effects on the metabolic phenotype of the colonic contents associated with reciprocal mucosal biomarkers of cancer risk. So somebody said earlier, it's never too early. It's actually never too late. Our results suggest that current guidelines of consumption of fiber-rich foods are too low, and increasing fiber to over 50 grams a day, which is what Dennis Burkert originally suggested in the traditional African diet, will in fact have a significant reduction effect on cancer in the USA and every other westernized population. They warn, however, against westernization of the African diet. We heard about that this morning, where reduction with westernization, it's becoming more common. Hamburgers at McDonald's, you see them in the cities in South Africa as well as Nigeria, I presume. Where in fact, if you suddenly go into a reduction in your traditional food intake with reduced beta-regenesis, that chronic inflammation might well be triggered into neoplasia. And with that I end. These studies are not possible by oneself. Our lab couldn't contain that, so we have to collaborate with, well, we love to collaborate with people overseas. Nicholson's lab at Imperial College to look at the metabolome, De Vos's lab in Wageningen, Holland, for the microbiota, and Gaskin's lab for the hydrogen-trophic organisms, and then finally funding from the NIH. Thank you.